starting, I've got a question for you. You all ever drank wine? I think that the answer is yes, and I'm going to show you that wine is a very important sector in the world. So this is a bottle of wine, and I'm going to show you that there are many things behind this. First, the producer. There are three big producers in the world, which are France, Italy, and the US. Just a little bit of numbers. France is producing 5 billion liters each year, and it represents 20% of the world's production. Aquitaine, the part of France which we come from, is producing 3% of the world's production. So it's very huge. Just to show you how wine is important in France, we are going to compare France and the US. So first, about the production. France is producing 155 liters of wine each second, and the US is producing 91 liters each second, so it's very huge. This production needs many people to be employed to produce it. So in France, 558,000 people are employed to produce wine, against 330,000 people are employed in the US. So this is very important. Then the economical aspect, just to show you the money that represent these sectors. In France, $17 billion are earned each year with wine sectors, and $14 billion are earned each year in the US. And last point but not least is the cultural aspect. So you all know that French gastronomy is very famous, and wine is a big part of this gastronomy. Indeed, French people are used to drink 1.5 glass of wine a day, whereas the Americans only drink quarter of a glass a day. So it's very huge. But something is threatening the sector, and this is downy mildew. So here you've got an healthy leaf, and this is a leaf affected with mildew. Mildew is also called Plasmopara viticola. It's an oomycete, which is a fungi-like microorganism. It affects the leaf, causing yellowish discoloration and oil spots. It acts by draining the plant's resources, avoiding it to accomplish its photosynthesis activities. Mildew spreads really quickly, and it can damage the vine. Indeed, Mag, which is a French magazine, published that in June 2015, some French vine growers lost more than 15% of their crops. Mr. Audebert, which is a vine grower from Bordeaux, testified that on its 24 hectares, this year more than 10 will be lost because of downy mildew, so it's very deplorable. There is a current treatment which is called Igem Bordeaux. It's made of copper sulfate. It kills mildew and it's very efficient, but it causes bad environmental issues. On the health, for example, through the water we drink that we take from the soil, it can cause severe stomach and headaches, and also have bad consequences on the environment, as copper is not degraded in the soil, and it's made bad problems on the ecosystem. So this is where the Igem Bordeaux team stepped in. This year we are composed of 23 students coming from the University of Bordeaux and an engineer school of biotechnology in Bordeaux. Our project is called Cured Vine, and it's aimed to find an alternative treatment to fight against downy mildew. So for the one who do not know where Bordeaux is located right there in France, in the Aquitaine region. So you may wonder what is our solution, and this is our solution. It's called Curdlan. It's a 1,3-beta-D-glycan, which is a glucose polymer. It's also a component of some pathogen cells walls, such as Agrobacterium tumefaciens. It brings many advantages because it's non-toxic, so we know that thanks to a collaboration with Oxford, and it's also biodegradable. It does not kill mildew as Igem Bordeaux does, but it stimulates the plant's defensive system. So you may wonder, how does it really act? And it acts like a vaccine for plants. So here you've got a cell, a leaf cell. So when we spray some curdlan on the cell, on the leaf, the cell recognizes the molecules and it leads to a depolarization of the membrane. The depolarization leads to a surexpression, a surproduction of H2O2 and salicylic acid. These two molecules are involved in the defense plants, the defense system of the plants. And once the stress is over, the plants reestablish its polarization membrane, but the molecules stay in the plants, and it acts like a wall which avoids interaction between mildew and the plants. And this is our solution. So now that you know how this curdlan works, let's present you how we did to produce it.
So thank you, Marie. Hello, my name is Charlotte, and I'm going to present to you the strategy that we had here at Igem Bordeaux. So what did we do? First, we looked at agrobacterium tumefactions, which is an organism which naturally produces beta-glucans. So you can see up here the synthetic pathway for production in agrobacterium. The problem with agrobacterium is that this metabolic pathway is only active in cases of stress. And in the laboratory, to stress the bacteria, it's commonly used through nitrogen starvation, which isn't necessarily easy to put in place in the laboratory. So what we wanted to do was to try to simplify this production by using different host organisms where we wouldn't need this nitrogen starvation. So we looked at 3 different host organisms to begin with. We rapidly eliminated Bacillus subtilis since it didn't have any of the required enzymes. E. coli we found had 3 of the 4 enzymes required, so we thought it would be interesting to try to insert the beta-1,3-glucan synthase, so the missing enzyme, in E. coli in order to have a beta-glucan production. And lastly, in yeast, we found that yeast naturally produce beta-glucans, but through a metabolic pathway that's not the same, so it doesn't have the same enzymes. And what we wanted to do in yeast was to try to overexpress the beta-glucan synthase in order to maximize curdlan production. So what we did was to look a bit at the genes which were involved in this production. So there are 3 genes in the operon, CRDA, CRDS, and CRDC. So CRDS is the gene which codes for the glucan synthase, which interests us. So we were particularly interested, and CRDA and CRDC help to translocate nascent polymer across different parts of the cell. So in E. coli, as I said, there's no natural curdlan production. So what we wanted to do was to take the gene from agrobacterium and insert it into E. coli, and to also use a promoter which is only active in stationary phase in order to maximize production. So we looked a bit at the Igem registry and found that in 2006, MIT characterized the osmoy promoter, which is indeed only active in stationary phase. So that's why we decided to work with that one. In yeast, so yeast naturally produce beta-glucans through the FKS1 gene, which codes for the glucan synthase. And as I said beforehand, we want to overexpress this gene, so we wanted to put in front of this gene the GAL1 promoter, which is inductible with galactose, in order to overexpress this gene. So we had 2 different strategies in yeast. The first one, we would isolate the gene by PCR and then insert it into a plasmid. We wanted to do a directed mutagenesis in order to get rid of the genes which were in the Igem plasmid, so that it will be compatible, and then clone our yeast with this plasmid. And our second strategy was to use homologous recombination to insert the promoter in front of the gene, and then use Gibson assembly to insert it into the Igem plasmid. So this year we managed to make several biobreaks. Unfortunately, we didn't manage to make our yeast biobreak, which is here present in the bottom right. We did, however, manage to make a biobreak with the GAL1 promoter and a histidine selection gene. We managed to clone separately the 3 genes which are useful for curdlan synthesis in agrobacterium, and to characterize once again the osmoy promoter from MIT in 2006, since it was out of stock when we needed it. And this allowed us to make our core biobrick, where we have the curdlan synthase gene and the promoter, which is only in stationary phase. So in order to figure out if we had managed to create curdlan or not, we chose to work with aniline blue. So So aniline blue is it's a substance that is specific of beta-glucans, and it has an absorption at 398 nanometers and an emission at 502 nanometers. So this allowed us to see if we had produced curdlan or not. So how did we proceed? We first transformed our E. coli strains. We cultivated them at 37 degrees until stationary phase, which corresponds to an OD of 0.8. Then we swapped the temperature to 27 degrees in order to maximize production. So you can see here a typical cell growth curve that we observed. And you can see now here the results that we had with the aniline blue staining. And so we can see here that the promoter that we chose to use does in fact work, since it's only when the bacteria are in stationary phase that the curdlan production begins to happen. So once we manage to produce our curdlan, we purify it. So this is done by lysing our cells with NaOH, multiple centrifugations. Curdlan is a substance which is soluble in alkaline solutions. So here at this stage, it's in solution. Then we precipitate it with acetic acid in order to purify it. And here we can suppose that the molecule is more or less pure. And then we quantify it with aniline blue, as I said beforehand. So what are the results that we had? We can see here in yellow the control and in green the modified strains. And so we chose to work in two different mediums, a minimal medium and a complete medium. And we can see that in both cases there's a significant increase in curdlan production. And overall, in 1 month, we managed to make 3.44 grams with only one gene of the operon. So, yeah, we found at this point, though, we were fairly surprised that we had a signal in the controls, and so we wanted to verify that the controls didn't have any curdlan. So we did this with a polarimeter. So we tested our controls and we tested the curdlan which we produced and compared it with the commercial curdlan which we bought. So we can see here that the optical rotation monitor is the same. It's fairly the same in our commercial curdlan and the curdlan we produced. So we can conclude that the molecule we produced is probably it's curdlan, and in the control it's not curdlan. So this is probably due to an aspecific aniline blue response. So then we tested our molecules on plants. We tested different cases without adding curdlan, with the curdlan that we produced, and with the commercial curdlan that we bought. We infected these plants with mildew, and then waited 6 days for the infection, and then we looked at the results. So the results that we got this week were when we didn't use any curdlan, there was 0% protection. So we found spores that looks like this on the leaves. And in our curdlan and with our commercial curdlan, we found that 85% and 95% respectively of the spores which we infected were no longer there afterwards. So we managed to produce a molecule which wasn't naturally present in E. coli, and we managed to verify that it does work on the plant. We only had 1 month, so we didn't manage to do everything that we wanted to do. It would be interesting to look at the optimization of the production, so this would be done by varying different parameters, particularly the stationary phase parameter, since that's when curdlan is produced. Our tests in yeasts didn't really work out since we didn't manage to isolate the gene and to clone it, so it will be interesting to look at that in further detail. And on a long term, we proved that there was an efficiency on the mildew, but maybe our molecule also has an efficiency on other pathogens, so that would be interesting to look at. And also to try to find the optimal concentration of curdlan to put on the leaf for safety and ethical aspects, which will now be talked about in our human practices section.
Hello, I'm Savandara. I will talk about human practices. As you know, human practices are an important topic in Igem competition. We have to ask one question: what do people think about synthetic biology? For us scientist students, it's something amazing, but it may be not the case for other people. So we created a survey that we submitted to random people in Bordeaux streets. The first question was, do you know synthetic biology? Around 70 people between 20 and 25 years old, 74% say no against 26%. So we have to explain what is synthetic biology because even the person who said yes can clearly define this field. After we give some example of application of synthetic biology, here you have an example look like to our Kidvine project. And after we ask them if they are for or against this application, here you have 75% are for or quite for this application. This survey is really interesting because it shows that synthetic biology is a field who people doesn't know anything about it, and popularization is very important. So we contacted Cap Science, which is a scientific and technical museum. We created with them Imagine Life project to popularize synthetic biology to the most of people. We also created a board game called Lab Life. It's focusing on biology and research. The goal is simple: to publish your first scientific paper, you have to create your own project. It's a funny way to understand scientific notion. Now, concerning our Kidvine project, we have to ask many questions on several ethical aspects. So we interviewed Mr. Jacques Fauché, a professor of bioethics from Bordeaux, and we asked him what is the definition of ethics in biology. As answer, he explained us the function of ethical committees and talked with them about several ethical aspects as intellectual properties or definition of living. We also met many people as winemaker in wine exposition and high school students to explain them our project. Finally, we organized a French meetup. Here you have all teams that have participated. During 2 days, we talked with each other about our project. We presented in front of a non-scientific public our project, and we had some debates on safety and ethics aspects. This meeting was very interesting because we can share with our team our question of synthetic biology. And now Marie will hand the presentation.
So we hope that we convinced you to try as much as we could to help the vines to fight against downy mildew. So we did this through the characterization of cyst biobricks, the carbon production, which is very efficient on plants as we show you, and the popularization of the project. So we want to thank Denis Dupuis, our supervisor, the crowdfunding participants, Cap Sciences, the museum, the IACB for lending us the laboratory, and our sponsor. So thank you for your attention, and if you have any questions, please ask.